Now, let's try to understand the bacterial cell structure further with a deeper discussion of the cell envelope. Prokaryotic cells are surrounded by complex envelope layers that differ in composition among the major groups. But first, let's talk about the basic structure of the bacterial cell wall in general, okay? We need to discuss this because a lot of antibiotics work on the cell wall. So let's say this is a bacterial cell. Now, outside the bacterial cell, we will primarily see that the cell wall is made up of sugar backbones. So these are also called glycans. When linked with adjacent glycans, they are now glycan chains. Now, these glycans have a chain of 4 amino acids that project out of each other. They are also called peptide or tetrapeptide links. And these peptides are cross-linked with each other with a peptide interbridge. So all of these components together make it the peptidoglycan cell wall. And these are all somewhat connected to each other to form one macromolecule of peptidoglycan. Now let's take one part of the cell wall and zoom in, just so we appreciate the structure even further. So the sugar backbones are actually made up of 2 monosaccharides, NAM and NAG. So NAM stands for N-acetylmuramic acid and NAG stands for N-acetylglucosamine. So these are connected to each other in beta-1,4 linkages and are in alternate configuration. Now each NAM has a set of 4 amino acids attached. So these are the tetrapeptide chains. This is one basic unit of the complex, which repeats all over around the bacterial cell. Now to make the bonds even stronger, the tetrapeptide chains are also cross-linked together with a peptide interbridge. Okay? So that is the basic structure of the peptidoglycan wall. This is also called murine or mucopeptide. So when you say murine wall or mucopeptide wall or peptidoglycan or cell wall, they are all synonymous with each other. Okay? So now, can you remember the single most important function of the peptidoglycan cell wall? Yes, it is what gives the bacteria its shape and structure, because if you remember, the bacterial cell wall does not have a cytoskeleton. But the most important function of the cell wall is to prevent the bacterial cell from undergoing osmotic burst. Okay? So how is this important for us to know? Remember, we have certain antibiotics that work by affecting the peptidoglycan cell wall. So it can be by damaging it or by preventing formation or synthesis of new peptidoglycan cell wall. Now, since you already have an idea of the structure of bacterial cell walls in general, let's talk about the rest of the bacterial cell envelopes, okay? So here we have 2 different bacterial cells. Both of them have similar bacterial cytoplasmic membranes. So these cytoplasmic membranes are similar to ours, no? In that they are made of bilipid bilayers. But one cell has a very thick bacterial cell wall outside the cytoplasmic membrane with up to around 40 sheets or levels of peptidoglycan. But the other kind of cell, however, has a very thin peptidoglycan layer, usually around 1 to 2 sheets only. But this kind, although it has a thinner cell wall, it has another extra bilipid bilayer outside the cell wall. Now this second or outer cell membrane is different from the first, the inner cell membrane. So let me tell you how. The inner side is still made of the usual line of lipids with the fatty acids, okay? But the outer side has different elements. The outer side is made up of a compound of a couple of sugars or disaccharides with phosphates, making a phosphorylated glucosamine disaccharide unit. Now, this unit has long-chain fatty acids also, which are actually unique from the fatty acids on the other side. No? So altogether, we call this unit lipid A. Okay? Now, lipid A has a corona or crown made of polysaccharide with a fancy headdress of repeat units. Now, the crown is called the polysaccharide core, and the headdress of repeat units are called the O antigen. Okay? So it is also important to take note that the polysaccharide crown or the core is similar in all species of this kind of cell. But the O antigen, the headdress, the repeat units, are actually unique in each species. Okay, so take note of that. Altogether, lipid A wearing their corona and the headdress is called lipopolysaccharide or LPS. It is also called endotoxin. Alright? Also, the O antigen is highly immunogenic, meaning it causes our immune system to react, but it is not toxigenic. Lipid A, on the other hand, is toxigenic, meaning it causes damage, but it is not immunogenic. Okay? Other endotoxins, however, only have shorter antennas instead of headdresses. If so, they are called lipo-oligosaccharides or LOS instead of LPS. Then also, the outer membrane is actually connected to the peptidoglycan layer by molecules of an unusual lipoprotein. This lipoprotein is also the most abundant protein in this kind of cell. But there is a space between the inner membrane and the outer membrane with a peptidoglycan layer in between. And this space is called the periplasmic space. Okay, now let's go back to the other cell. Now, this cell doesn't have feathers or fancy headdresses, but it has long, curly antennas that span the cell wall, and which are made up of glycerophosphate or ribitol phosphate residues, and they are called teichoic acids. Now, there are two types of teichoic acids, no? Those that extend from the membrane or membrane teichoic acids or MTA, and those that extend from the peptidoglycan only, which are called wall teichoic acids or WTA. Now, these curly antennas or teichoic acids are important for antigenic determinants or attachment factors of the bacteria. There are also other curly antennas that are made up of sugar units instead of phosphoric units, and we call these teichuronic acids. Okay? Now, if you take both bacterial cells that we have drawn and we stain them using a specific stain called the Gram stain, this cell would stain blue and the other cell would stain red. Okay? So this cell with the thick peptidoglycan layer will stain blue. This is actually the Gram-positive bacteria. The other cell, this other cell with the outer layer of cell membrane is the Gram-negative cell. By the way, another thing I need to add, peptidoglycan is a very porous layer, so it is highly permeable. The outer membrane of Gram-negative bacteria, however, are not as permeable, so to allow passive diffusion of low molecular weight hydrophilic compounds, it has special channels called porins. This is also important to know because this actually accounts for the relatively high resistance of Gram-negative bacteria to some antibiotics, because large antibiotic molecules are able to penetrate very slowly. Alright? The number of porins also differ among different species. For example, Pseudomonas species have a very few number of porins. That's why this makes them even more resistant to many antibiotics. Okay?